महानिदेशक महोदय, उप महानिदेशक महोदय, प्रशासन प्रभारी और मेरे सारे प्यारे साथियों, मैं डॉक्टर ऋतिका हसीजा नरूला, अनुसंधान अधिकारी, केंद्रीय होम्योपैथी अनुसंधान परिषद की ओर से आप सभी का आज के इस एक दिवसीय रोगाणुरोधी प्रतिरोध जागरूकता अभियान वन डे एंटीमाइक्रोबियल रेजिस्टेंस अवेयरनेस कैंपेन में हार्दिक स्वागत करती हूँ। ये अभियान विश्व एएमआर जागरूकता सप्ताह के उपलक्ष्य में आयोजित किया जा रहा है। प्रतिवर्ष 18 से 24 नवंबर के बीच विश्व एएमआर जागरूकता सप्ताह यानी वर्ल्ड एएमआर अवेयरनेस वीक मनाया जाता है, जिसका उद्देश्य शिक्षा, प्रशिक्षण एवं संचार के माध्यम से एएमआर संबंधित संवेदीकरण तथा जागरूकता फैलाना है। The theme for the World AMR Awareness Week this year is Educate, Advocate, Act Now. AMR occurs when bacteria, viruses, fungi and parasites no longer respond to the antimicrobial medicines. AMR is a natural process that happens over time through genetic changes in pathogens. Its emergence and spread is accelerated by the human activity, mainly the misuse and overuse of the antimicrobials to treat, prevent or control infections in humans, animals and plants. It is a grave and growing threat to the global health, one that endangers our ability to treat common infections, perform surgeries and even manage complex health conditions. AMR is not solely a matter for conventional medicine to solve. We must broaden our perspective and look at the complementary and the integrative approaches, including homeopathy, to tackle and alleviate the crisis of AMR. Today, we have gathered to learn more about various aspects of AMR and explore the role of homeopathy in combating this global concern. Homeopathic approach, as we all know, is not about killing microorganisms. It seeks to promote and prevent patient resistance to infection, modulate innate immunity, and cultivate a healthy microbiome. An effective, safe, evidence-based and less invasive treatment is the need of the hour, and the current scenario of AMR, which is indeed offered by homeopathy, which we know is highly acceptable, relatively safe, and cost-effective. And the most important thing, that there is no risk of developing resistance with these homeopathic medicines, since these medicines are not pathogen specific. They don't work upon any specific strains like the conventional medicine. AMR एक गंभीर समस्या है जो न केवल भारत में अपितु पूरे विश्व में चिंता का विषय बन चुकी है। केंद्रीय होम्योपैथी अनुसंधान परिषद का ये प्रयास है कि होम्योपैथिक शोध एवं अन्य रचनात्मक गतिविधियों के माध्यम से इस क्षेत्र में कार्य किया जाए। अब इस कार्यक्रम का शुभ आरंभ करने के लिए मैं हमारे महानिदेशक महोदय डॉक्टर सुभाष कौशिक जी, जिनके मार्गदर्शन और नेतृत्व ने आज के इस कार्यक्रम को आकार दिया है, उनसे अनुरोध करती हूँ कि आप हमारे सम्माननीय वक्ताओं का पुष्पों द्वारा स्वागत करें। पहले डॉक्टर योगेश एस शाउजे जी, डायरेक्टर स्कैन रिसर्च ट्रस्ट बेंगलुरु एंड फॉर्मर साइंटिस्ट NCCS पुणे महाराष्ट्र। We welcome you, sir. Dr. Sonal Saxena, Director, Professor and Head, Department of Microbiology, Maulana Azad Medical College. Please give them a big round of applause. We welcome you, Madam, sir. Thank you, DG sir, for welcoming our speakers. So we are welcoming our first speaker, Dr. Yogesh S. Saujiji, Director, Scan Research and Former Scientist, NCCS Pune. Dr. Sonal Saxena, Director, Department of Microbiology, Maulana Azad Medical College, New Delhi. Thank you, sir. Now I would request our Director General, sir, to deliver the keynote address on AMR.
ये माइक थोड़ा सा ऊंचा करना ठीक है इट्स फाइन ठीक है A very good morning everyone and a warm welcome to respected Dr. Yogesh S. Saucheji, Dr. SKN Research Trust Bengaluru, and Dr. Sonal Saxena ma'am, Director, Professor and Head, Department of Microbiology, Maulana Azad Medical College, New Delhi. Thanks return to both of you for accepting our invitation at such short notice. We are obliged. Today, we all have gathered here to address one of the most pressing health challenges of our time, that is antimicrobial resistance. This AMR, as we all know, that antimicrobials are critical tools for fighting diseases, not just in humans, but also in animals and plants. They are the cornerstones of modern medicine. The emergence and spread of drug-resistant pathogens threaten our ability to treat common infections and to perform life-saving procedures. It is a great concern that these medicines are becoming ineffective, thus posing potentially catastrophic risk to global public health. The misuse and overuse of the antimicrobials in human, animals and plants are the main cause of drug-resistant pathogens. The lack of clean water, sanitation and hygiene, and inadequate infection prevention and control promotes the spread of microbes resistance to antimicrobial treatment. According to World Health Organization, AMR could lead to 10 million deaths annually by 2050. These statistics are alarming, but they are also serve as a call to action for all of us. AMR affects countries in all regions and at all income levels. Its drivers and consequences are exacerbated by poverty and inequality. And low and middle income countries are most affected. The world faces an antimicrobial pipeline and access crisis. In addition to death and disability, AMR has significant economic costs. The cafeteria approach to AMR offers a promising framework for combating this global growing health threat by combining a range of interventions and tailoring them to specific needs. We can work towards a more sustainable and effective response to antibiotic resistance. Empowering the patients to choose the treatment approach while informing them about the pros and cons of each system of medicine for particular disease will improve the acceptability of medical interventions and patients' compliance to treatment. Integrative medicine clinics in all parts of the country must be promoted, where doctors from all the system of medicine sit together and offers a choice for the patients. As today, as we all know, that is not a time for competition, rather it's a time for cooperation. And also, we all have to follow the recent mandate for integration by our government. The growing global threat of antimicrobial resistance poses significant challenges to conventional medicine, particularly in treating infections that were once easily managed with antibiotics. And now, AMR continues to limit the effectiveness of the traditional antibiotics. Considering these challenges, homeopathy offers a promising alternative and can play a crucial role in combating the AMR crisis. An effective, safe, evidence-based and less invasive treatment is the need of the hour in the current scenario of AMR. That is where homeopathy takes a lead. Clinical research data has demonstrated the potential of homeopathy in treating a wide range of diseases, offering benefits not only in symptom management but also in improving overall patient outcomes. Clinical studies have demonstrated the effects of homeopathic medicines as antibacterial, antiviral and antifungal agents. Furthermore, the role of homeopathy as an adjunctive therapy in treating multi-drug resistant infections has also been evaluated. However, there is a need to replicate and undertake more of such studies. It has also been observed that in homeopathy, if homeopathy is used in the early stages of URTI treatment, particularly during the initial phase, we can reduce the use of antibiotics by up to 75%, which can significantly help in combating antimicrobial resistance, and we have published data to authenticate this fact. In the National Action Plan of AMR 2.0, we have proposed for the inclusion of Ayush intervention, especially homeopathy, at almost all the strategic level of this action plan. Sir and Madam, I just want to tell you that under the aegis of Ministry of Ayush, CCRH is an apex research organization which undertakes, coordinates, promotes, develops and disseminates scientific research in the field of homeopathic system of medicine. CCRH basically engaged in multi-dimensional research activities, like to start with survey and collection of medicine plants, drug standardization, drug proving, clinical research, clinical validation, epidemic research, literary research, even public health programming research mode and so on. As we were talking upstairs, that CCRH undertake these different research studies through different institutes and units spread across the country, and they are 35 in numbers, where specific research studies are conducted along with the clinical care laboratory facilities, and we are also having IBD facilities at some selected centers. So we are also engaged in basic and fundamental research in homeopathy, conducted on biological models in vitro and in vivo, and on physico-chemical models. We also undertake collaborative research with collaborations of many national institutes of repute, having the expertise and infrastructure to validate the concept of homeopathy on scientific parameters. In the context of AMR, CCRH signed a letter of intent with ICMR NIRT, Chennai, to undertake preclinical evaluation of homeopathic drugs, studies on MDR strains of tuberculosis for a group of homeopathic medicines. CCRH has also collaborating with institutes like IVRI, Bareilly and Duvasu, Mathura, to undertake veterinary studies in the AMR, and results have been encouraging for homeopathic intervention in diseases like neonatal calf diarrhea and mites-induced dermatitis in dogs. In the field of agro, CCRH has collaborated with the College of Agriculture, Professor Jayashankar, Telangana State Agriculture University, Hyderabad, wherein the efficacy of homeopathic drugs was evaluated for their antifungal activities against Sclerotium ralsii, yielding positive outcomes. Leads of homeopathy from the evident literature may be taken, and an awareness may be created among all stakeholders for adopting homeopathy, especially in the initial stage of all infections as a standalone or as an add-on to standard care. So on the occasion of the World Awareness AMR Week, which is held every year between November 18 to 24, it gives me immense pride that CCRH has taken this initiative to organize this one-day campaign, which goes in synchronicity with the theme of this year's AMR Awareness Week, that is Educate, Advocate and Act Now. This year's theme calls on the global community to educate stakeholders on AMR, advocates for bold commitments and take concrete actions in the response to AMR. Stronger political leadership, advocacy and accountability are needed at all levels, and at the time of act is now. At CCRH, we are committing to bring at the forefront the fight against AMR. We have organized this campaign not just to raise awareness but to mobilize action. I urge each of you to take the knowledge you gain today from these two eminent personalities and share it within your networks. We have a unique opportunity to create a ripple effect and awareness that is spread from individual actions to community. Wide range. Remember, every conversation you have about AMR is a step towards a healthier future. I am pleased to announce that various activities, including a quiz, poster competition and a slogan contest, are being organized to spread the awareness about AMR. These engaging activities will provide an interactive platform for everyone to contribute to the cause and deepens our understanding of this critical issue. I would like to extend my sincere thanks to our experts who have contributed to this awareness program. I am confident that today's session will provide us with a new direction and further strengthen our efforts in combating AMR. As we embark on this day of learning and engagement, let us remember that combating AMR requires a united front. Together, we can foster a culture of responsibility and awareness that will help safeguard our health system and protect future generations. Thank you for your dedication, your presence, and your commitment to making a difference in the fight against antimicrobial resistance. Let us make this campaign a turning point in our collective efforts to address this global concern. Thank you. Thank you very much.
Thank you, sir, for your insightful words. Your address has set the tone for this important discussion today. अब मैं CCRH के उप महानिदेशक डॉक्टर सुनील रामटेके जी को आमंत्रित करती हूँ कि वह होम्योपैथिक पर्सपेक्टिव इन AMR पे अपने विचार व्यक्त करें।
सभी को मेरा नमस्कार। Respected dignitaries, Dr. Subhash Kaushik ji, Director General, Central Council for Research in Homeopathy, Dr. Yogesh Sauche ji, Director, SCAN, Bangalore, Dr. Sonal Saxena, Director and Head of the Department, Microbiology, Maulana Azad Medical College, Delhi, Dr. Renu Mittal, Scientist for CCRH, officers and officials of CCRH. It's our pleasure and privilege to have presence of respected eminent speaker and guest of honors, Dr. Yogesh Sauche ji and Dr. Sonal Saxena. First of all, on behalf of CCRH, with great pleasure, I would like to welcome respected dignitaries, speakers, officers and officials of CCRH to the one-day awareness campaign on antimicrobial resistance, which is being organized today on the occasion of World Antimicrobial Resistance Awareness Week. I really would like to appreciate all the distinguished guest speakers for accepting our invitation to share their knowledge and vast experiences in tackling AMR. CCRH is conducting this today's program to create awareness about AMR by organizing expert talk, quiz competitions, slogan writing competition, and poster competition. Antimicrobial resistance is an urgent global public health problem which increasing day by day worldwide. AMR is a natural process that happens over the time through genetic changes in pathogenic organism. Its emergence and spread is accelerated by human activities, mainly by misuse and overuse of antimicrobial to treat and prevent and control infection in human and animal and plants. We all know that whenever we fall sick, we directly approach to the pharmacy and we are taking the drugs over the counters, despite of knowing which infections we are having, whether we are having bacterial infections, viral infections, or parasitic infection or fungal infections, despite of knowing that we are going in pharmacy and we are taking the medicines. Even when the patient approaches to the hospital in emergency, the doctors used to prescribe at a time antiviral, antibacterial, antifungals, antiparasitic even at a time to the patient, despite of knowing the patient which type of the infection his patient is suffering from. The AMR occurs when the microorganisms such as viruses, bacteria, fungi and parasites no longer respond to the antimicrobial medicines. हम सभी जानते हैं कि ये माइक्रोबियल रेजिस्टेंस कैसे होता है. जब हम कुछ एंटीबैक्टीरियल लेते हैं, पर्टिकुलरली मोस्टली ज्यादातर लोग एंटीबायोटिक लेते हैं बैक्टीरिया को किल करने के लिए. तो बैक्टीरिया को किल करते करते कुछ तो बैक्टीरिया मर जाते हैं, कुछ बैक्टीरिया उस एंटीबायोटिक को रिसीव करते हैं और वो उनका जो पावर जो है उसकी जितने बार आप एंटीबायोटिक लोगे उतने बार जो बैक्टीरिया अलाइव रहते हैं वो डे बाय डे क्या होते हैं, स्ट्रॉन्ग हो जाते हैं. जैसे मैं आपको एक एग्जांपल बताता हूं कि किसी को अगर सांप काटता है, स्नेक बाइट होता है और वो सरवाइव करता है, उसको फिर दोबारा काटता है. मैंने खुद हमारे गांव में इस तरह का केस हुआ है. तो उसको बहुत बार स्नेक काटता था लेकिन उसको कुछ नहीं होता था. He developed the resistance. वो बार बार उसको काटने से. तो इसी तरह हमारे बॉडी में बहुत सारे माइक्रोब्स होते हैं जिसको माइक्रोबियम बोलते हैं हम. और वो जो माइक्रोबियम जो होता है वो हमारे बॉडी में जो हमारे जो हेल्थ को मेंटेन करता है, हमारे वेलबींग को मेंटेन करता है. हम यानी उससे हमारे इम्यून सिस्टम को बूस्ट अप करता है. इवन हमारे जो जितने भी हमारे शरीर में जो ऑर्गन्स है उनके फंक्शन के लिए हेल्प करता है. हमारी जो हार्ट है, स्टमक का फंक्शन है, जो डाइजेशन है, वो सारा कुछ वो जो माइक्रोबियम जो है वो कंट्रोल करते हैं, हमारी इम्यून सिस्टम को मेंटेन करते हैं. तो इसमें हमें इसको अभी कैसे प्रिवेंट करना है. अब एंटीबायोटिक क्या होती है, एंटीबायोटिक नई एंटीबायोटिक हर 5 साल में 10 साल में वो चेंज होती रहती है एंटीबायोटिक. अब इसको एंटीबायोटिक जितने मर्जी आप आ जाए और कुछ दिन के बाद क्या होता है, वो पेशेंट क्या है रेजिस्टेंस उसमें डेवलप हो जाता है और वो एंटीबायोटिक काम नहीं करती. जैसे अभी माइक्रोबैक्टीरियल ट्यूबरकुलोसिस में रिफाम्पिसिन काम नहीं करती है. तो वो रेजिस्टेंस उसमें डेवलप हो जाता है. अब इसको एंटीमाइक्रोबियल रेजिस्टेंस को प्रिवेंट करने के लिए हमारा काम है प्रिवेंटिव मेजर्स को अपनाना. इन्फेक्शन को ही नहीं होने देना चाहिए बॉडी में. जब हम इन्फेक्शन को नहीं प्रिवेंट करेंगे बाय वेरियस मीन्स, हमारा अपना सैनिटरी हाइजीन्स मेंटेन करना है, सैनिटेशन मेंटेन करना है, अच्छे क्लीन क्लोथ पहनना है, घर में स्वच्छता का पालन करना चाहिए. घर में अपने बार कहीं पर जो मार्केट में जो ओपन जो फूड होते हैं, जो ओपन खाना होता है उसको नहीं खाना चाहिए. तो तभी और हमारी जो बार कहीं पर भी गए तो बिना हाथ धोए कोई भी चीज नहीं खानी चाहिए. अच्छा. तो तभी हम AMR को प्रिवेंट कर सकते हैं. तो गवर्नमेंट ऑफ इंडिया हैज गिवन due cognizance to the problem of AMR, and to tackle this issue launched a national program on AMR containment during the 12th 5-year plan, that is 2012 2017, which is being coordinated by National Center for Disease Control, Directorate General of Health Services, Ministry of Health and Family Welfare. The main objective of this program are to establish lab-based AMR. Lab-based AMR surveillance system in the country to generate quality data on AMR. Carry out surveillance of antimicrobial uses in different healthcare settings. Strengthening the infection control practices and promote rational use of antimicrobials through antimicrobials stewardship activities. Generate awareness among healthcare providers and community on AMR and rational use of antimicrobials. Now, हम सभी जानते हैं कि हमारी जो होम्योपैथी में एक बहुत बड़ा एक स्कोप है. हम बिना डायग्नोसिस से अर्ली स्टेजेस में पेशेंट को हम ठीक करते हैं. It is the great scope in homeopathy that homeopathic physician can treat the disease without naming the disease or before arriving at confirmed diagnosis on the basis of symptom similarity. This is the great scope in homeopathy, and by which this we can prevent various types of infection, various types of infectious diseases during the initial period, initial stage of the infections. And CCRH has conducted many trials on upper respiratory tract infections and observed the effectiveness of homeopathy. By using this homeopathic medicine as an add-on in various trials, we can reduce the hospital stay. We can even reduce even we can prevent the patients from loss of their wages. So these are the benefits of homeopathy, and many things I have written here, but earlier our DG sir and even Dr. Ritika covered the majority of the parts of my talk. Therefore, I am not going to repeat it again. Now, the CCRH has proposed an action plan for combating AMR, which include the development of public use guidelines of homeopathy based on preventive measures for behavior change to prevent microbial infections, under which we would promote awareness campaign among general public and use of homeopathy in prodromal or early phase of infection to combat it early and permanently. Preventing infections through effective sanitations, hygiene and infection prevention measures like immunization, safe food preparations, hand washing, maintaining clean clothing, clean house, clean surrounding and avoiding taking open food, I already told, and using antibiotic as directed and only when necessary. Promoting use of homeopathy in epidemic diseases, which is not only a safe but also effective and cost effective. the development of guidelines for homeopathic practitioners for management of specific and common microbial infections. Under the training and capacity building of homeopathy practitioners and paramedics for promoting the use of relevant homeopathic intervention in infectious diseases and AMR issues. The development of medicine kits and training manuals for homeopathy practitioners under the parameters of innovations, research, interventions in respect of developing alternatives to antimicrobial and adjuvant remedies for infectious diseases. We have proposed to undertake research studies, preclinical and clinical trial for testing efficacy of homeopathic medicines in modulating resistance toward antibiotic both in vitro and in vivo models with ICMR, NIRT. We are in the process of initiating the preclinical study on MDRI tuberculosis. Let us stand united in the mission, taking a comprehensive, holistic approach that integrates the best of conventional and complementary medicine to combat antimicrobial resistance and safeguard global health. I am sure that you all will feel enriched with the knowledge by participating in today's program and attending expert talk from learned, eminent speakers. I express my sincere gratitude to respected Director General CCRH and Dr. Ritika and her team for giving me opportunity to speak on this occasion. I would like to thank Dr. Ritika for taking initiative to organize and coordinate today's program. Thank you, Ritika. I greet the grand success for this today's program. Once again, welcome one and all to the today's program. Have a nice day to one and all. Dhanyavad.
Thank you, sir, for enlightening the audience with the homeopathic perspective, and thank you for your kind words. Now, I would request all the dignitaries on the dais to grace a brochure on homeopathy for AMR, which was released on the occasion of World Homeopathy Day to spread awareness among people. Esteemed speakers to grace our brochures. This is already released, ma'am. We will all break for a cup of tea. We will reconvene in half an hour's time for the sessions of the day. I request all to gather for the high tea, please. We will begin with our sessions after the tea break. So welcome back everyone. I hope you had a refreshing tea break. Our first guest speaker for the day is Dr. Yogesh S. Chowche. He is currently working as director at SCAN Research Trust, Bangalore. He started his career as scientist, National Center for Cell Sciences in Pune. He's working since 25 years in the field of microbial ecology, microbial molecular taxonomy and biodiversity. He has more than 400 publications to his credit. He is also an editorial board of prestigious journals like Current Science and PLOS ONE. During his tenure at NCCS, he established the Microbial Culture Collection, MCC, now known as National Center for Microbial Resource. It holds more than 150,000 bacteria and fungi in its collection, recognized by the World Federation of Culture Collection. MCC is also recognized as International Depository Authority and is designated repository by Ministry of Environment, Forest and Climate Change, Government of India under the Biological Diversity Act 2002. In September 2018, Department of Biotechnology extended the mandate of this repository to include antimicrobial resistant microbes. I feel proud and honored to announce that as a recognition of Dr. Chowche's contribution in the field, a genus, Shaushella, has been named. In his honor, a big round of applause to have such an eminent speaker amongst us today. Interestingly, after recent reclassification, this genus now contains well-known probiotic species, Bacillus clausii is now Shaushella clausii. Today, sir will be deliberating a big round of applause. Today, sir will be deliberating on the topic, growing menace of AMR and need for its mitigation. Over to you, sir.
Good morning to all of you, and thank you very much for Honorable DG and Dr. Ritika for inviting me here. And it's indeed a great pleasure to talk to you today about growing means of AMR and the need for its mitigation, because this is a topic which is close to everybody's heart, and as it was mentioned in the earlier part of the day, that everybody has to work towards it. Fortunately, many of the things which I wanted to say, a passing reference to those has been made in the earlier part of the day, so my job has become easier, and I'll just elaborate on some of the things that were mentioned in the earlier part of the day. So, actually, antibiotic resistance is not new, and it was there even before antibiotics came. So we somehow, it has become important and dangerous because of the indiscriminate use of antibiotics. But if you look at historically, antibiotics were there even before commercial use of antibiotics started. So there have been a lot of studies where people tried to look for the presence of antibiotics and antibiotic resistant genes. So they have found out antibiotic resistant genes and antibiotics in mummies and even in pristine environments. So both antibiotics and antibiotic resistance are there in nature several years ago. So it's not a new thing, but as was said earlier, and as we know that the problem has become acute because of the indiscriminate use of antibiotics when the commercial use of antibiotics started at the time of Second World War with the discovery of penicillin. But the problem has now reached to alarming proportions, and it is estimated by 2050 there will be 10 million deaths because of the AMR costing something like 66 trillion pounds, and every 3 seconds one person will die because of the antibiotic AMR, and the deaths due to AMR, that is antibiotic resistant infections, would be more than the death of the cancer. So like pre-antibiotic times, see, I mean, if we again go back in the history, before the discovery of antibiotics, the major cause of death was infections. So even with a simple, I mean, there are examples where somebody had just was injured by a thorn, and then that developed an infection and the person succumbed to the infection. So it's a fear that we'll go back to the same era, because a simple wound get infected by antibiotic resistant organism resulting into the death. So there is a fear that we will go back to the pre-antibiotic era if this threat of AMR is not controlled. And it is all over the world. So if you look at the global scenario, no country is exception to that. For different reasons, the threat of antibiotic resistance then in globally all over, it doesn't discriminate between a developed country, developing country, and underdeveloped country. It doesn't discriminate that this way or that way. The problem exists all over the globe. So it's a very serious problem, and we have started to realize the severity of the problem now. Now, just though it was mentioned in the earlier part of the talk, just to recapitulate, basically antimicrobials or antibiotics are the agents that are used to kill infectious organisms, and depending on the nature of the infectious organisms, there are different categories. You have antivirals that act against viruses, which are very difficult to get actually, because viruses use host cell for all their metabolism. So very specific antiviral is very difficult to get. Then you have antiparasites, you have antifungals, which are used for typically for fungal infections, which include skin infections and systemic infections also. And broadly, when we talk about antibiotics, these are basically antibacterial. So the agents that are active against antibacterial. However, the problem of resistance is not there just for antibacterial. The problem exists for antifungals, because there is a rise on antifungal resistant skin infections. There has been a rise on that. There are increases incidences of antiviral resistances also, and antiparasite resistant. We all know drug resistant malaria. So the problem of antibiotic exists in all the antimicrobials, whether it is antiviral, antiparasites, antifungals, or antibiotics. So the problem exists everywhere, and it's equally serious problem for fungal and viral infections as well. So also malaria. Since we are going to talk more on antibacterial than any other organism, just a quick summary that there are a variety of antibiotics. Antibiotics belong to different classes: sulfonamides, then fluoroquinones, tetracyclines, aminoglycosides, macrolides, and a group of penicillins. And they have different targets. So penicillin, historically, as we all know, that it inhibits the synthesis of cell wall. Fluoroquinones inhibit DNA replication. Sulfonamides and trimethoprim, they inhibit folate synthesis, and tetracycline aminoglycosides, they inhibit protein synthesis. Besides that, there are some antibiotics now which inhibit fatty acid biosynthesis in bacteria, and I'll just make a brief mention of that later part of my talk. So the target of different class of antibiotics is different, and obviously then the mechanism of resistance for these each class is different. So basically how the antibiotic resistance develops is that because of the exposure to the antibiotic, the organism, there is a genetic change in the organism leading to the resistance, and then due to suboptimal concentration of antibiotics, these resistant organisms survive and then they spread. That is basically the mechanism. So it is basically a genetic change. And the serious problem is that in bacteria is that many times this change resides in the extra chromosomal element in bacteria. So since it resides in the extra chromosomal element that can be easily transferred from one bacterium to the other bacterium, the resistance spreads fast because the extra chromosomal elements are transferred faster in bacteria, so the resistance also spread fast, and also many of these elements do not recognize species and genus boundaries. So that's why the resistance spreads across the species and genera, and which is a very, very serious threat. So resistance developed in one organism can spread to any other organisms because of this lateral transfer or horizontal gene transfer as it is called. So at the molecular level, the mechanism acts by different mechanisms. I'll not go into the details of mechanism for each and every antibiotic, but if you want to classify them, in some cases there is a modification of drug target, like for example penicillins, there is a cleavage of the molecule. In some cases the drug gets chemically modified in the resistance mechanism, as in the case of chloramphenicol, there is a chemical where there is chemical inactivation of the drug moiety. In many cases it is active efflux of the drug, so drug that comes inside the cell is actively thrown outside the cell, and in some cases the intake of drug is limited. So if the drug is taken inside by active transport, that uptake is limited. So these are the broad mechanisms of development of antibiotic resistance. In some cases, I have mentioned all the possible mechanisms, and so this again summarizes in pictorially how various mechanisms work. So it basically says the same thing, that in some cases there is alternative target which is called as target bypass. So and in some cases there is upregulation and downregulation of antibiotic efflux. So there are various mechanisms by which the antibiotic resistance develops, and most, if not all, are the genes for these reside on extra chromosomal elements. So this particular resistance is passed across the species and across the genera. So in an environment, then how this antibiotic resistance spreads? So basically it is the indiscriminate use of antibiotics and indiscriminate usage, disposal of waste that is responsible for the spread of the antibiotics. So historically again it happened through the accidental discovery, but somewhere during the Second World War itself, the use of antibiotics for mass production of animals was started. So it was initially started because it is economical to, whether it is poultry or cattle, it is economical to put a large number in a small space for economical purposes, and then obviously if you give them antibiotics, the infections will be less and the productivity is more. So as a side effect of that, it was also discovered that the antibiotic treated poultry gains weight faster, and so the indiscriminate use of antibiotics in poultry was started, and then that was the greatest way by which antibiotic resistance spread dramatically. And of course the waste was not monitored, so waste from cattle industry, waste from poultry industry was not monitored. So then the resistance spread everywhere, to soil, to water. Even, I'm not going to present that part of our work because it is quite old, but even the antibiotic producing factories, if their waste is not disposed properly, again that goes everywhere. That goes to soil, that goes to well, that goes to rivers, and that again leads to spread of antibiotics. So that's how from one source to another, from cattle, from soil, from water, the antibiotic spreads everywhere. Even the hospital waste is another source of spread of antibiotic resistance in the environment. So here it is just shown with one example as to how one person can spread the antibiotic resistance everywhere. So the person spreads to community, person spreads to environment, and that's how the antibiotic resistance, a single person can spread antibiotic resistance to across the population by coming in contact with everything that it comes across through its feces, through the food and many other sources. The antibiotic resistance from one person can be spread to hundreds of other people. So considering this threat, since this threat was realized recently and a lot of work started on that, the different category of infections agents are resistant to different groups of antibiotics, and the severity of the infection also varies. So considering all these parameters, WHO came with this priority pathogen list. This is a WHO list, and these are the pathogens listed here are they have classified these organisms as critical, high and medium level importance, and this is the WHO list of priority pathogens in terms of AMR or antibiotic resistance. So the topmost priority is Acinetobacter baumannii, carbapenem resistant Acinetobacter baumannii, carbapenem resistant Pseudomonas aeruginosa, carbapenem resistant and third generation cephalosporin resistant Enterobacteriaceae. So these are the highest pathology, highest category, and then followed by second priority and medium category. But the situation in India is different. What is observed globally or in the Western world is not true for India. Some of the organisms which are important for India are not important globally and vice versa. So a couple of years back, Department of Biotechnology and WHO India office came together with this what is called as Indian priority pathogen list, and this is a list which is important for India, and that again categorizes pathogens in three categories: critical priority, high priority and medium priority. And if you do a comparison, the organisms in this list are different. So in India we have a different Indian priority pathogen list, and this keeps on changing. I mean, depending on the need of the time, the list gets revised, but presently this list was prepared around 2018. This is a list which is presently being used in India. So what are the factors that contribute to antibiotic resistance? I'll quickly go through this. So these are drug-related factors, environmental factors, patient-related factors, and prescription-related factors. So I'll quickly summarize those. We all know about that. So I have already mentioned that environmental factors include the widespread use of antibiotics in animal industry, poor sanitation, and ineffective infection control programs. Because as was mentioned earlier in the day, that if there is a proper infection control, then there will be no infections and the need for antibiotics will be less. This is the patient-related factors is a very serious problem in India. It is my personal experience that even in educated class there is a self-medication and there is also a not compliance to the dosage. So when I was in NCCs, I have always seen this, that even the doctoral students, if they are not feeling well, they will go to a chemist and get antibiotic themselves and start treating themselves, and they would not complete a proper dose. After they start feeling better, without bothering to complete the dose, they will stop taking antibiotics. So this is a problem with educated class also. So this is a very serious problem in India. Availability of antibiotics across the over the counter and not completing the dose. This is a very, very serious problem in India. And of course, we all know that indiscriminate use of antibiotics. So if you go to a doctor without checking for though many hospitals have a antibiotic policy, but still in many hospitals, without waiting for the test report, the most broad spectrum antibiotic is prescribed. And then that creates a problem, because if there is a resistance to that antibiotic, then there is nothing left. So if the patient gets some other infection, there is nothing left. And there have been cases, many cases where because of this problem, when patient really had infection, whether it was a wound infection or some other kidney infection, the patient was resistant to every possible known antibiotic. So there was no line of defense was left, because earlier in the life, the patient was given antibiotics indiscriminately, and probably he did not follow the proper dosage. So this is a really very serious problem in the country. And fake drugs. We all recently, the fake drug racket was in the news. So fake drug, the drug which are not of sufficient potency, because if you get a sublethal concentration of the drug, then that develops more serious antibiotic resistance. So these are all the problems which are responsible for the development of antibiotic resistance. And now with this, again, this was Dr. Ramteke mentioned this during this talk, and I'm glad that he made a mention of that. So I'll talk to some of the issues related to antibiotic resistance which are very close to us. So antibiotic resistance is a problem not only when we get infection. We have antibiotic resistant organisms within us. So all our parts of our body, whether it is gut or skin or oral cavity, we harbor large number of microbes. So the total number of microbial cells in our body is as much as our own cells. So cell number wise they are equivalent, and in terms of genetic DNA, their microbial cells contribute much more. And these being microbes, they also carry antibiotic resistance. So even if you do not take any antibiotic, your gut would harbor antibiotic resistant bacteria, and indiscriminate use of antibiotics makes it worse, because as I mentioned earlier, that there is a horizontal gene transfer, indiscriminate horizontal gene transfer in bacteria. So you have infection from antibiotic resistant organism, you do not take antibiotics properly, but then those genes get transferred to some of your commensal bacteria, and then when you get some other infection, these genes go from commensal bacteria to the new pathogen, and thus you land up into more serious problem. So the antibiotic resistance in commensal organisms in our body is also equally serious problem on one hand, but on the other hand, it also offers you a solution for an alternative treatment. Because I'm not going to talk about that, but because there are good commensal bacteria, one of the therapies that is being used currently for the most antibiotic resistant infection, which is called as Clostridium difficile diarrhea, is use of good commensal bacteria. So they do this by fecal microbiota transplantation, and it has shown 100% success abroad. The infection is not very common in India, it is more common abroad. So this being an antibiotic resistant infection, hospital acquired infection, the mortality was almost 100%. But with fecal microbiota transplantation from a healthy donor, the mortality came down by 99%. So survival increased by 99%. So the understanding of healthy gut bacteria has given us a tool one of the tools to combat antimicrobial resistance. But as I said, that it is naturally there, and I will not talk about global scenario. There are lot of global studies which talk about naturally occurring antibiotic resistance in the commensal organisms on our body. I'll just show you our own example. So when I was in NCCS, we had initiated this Pan-India Human Microbiome Initiative, which is currently led in NCCS by my colleague Dheeraj Dhotre, and it is a multi-institutional project across the India. Many institutes were involved in this, funded by Department of Biotechnology. And what we found, this was a pan-India study, so we studied 4 biogeographic regions and 17 populations were studied here, and a total 3,400 individuals were studied, 200 individuals from each group. So 17 communities were studied. And what we found interestingly was the overall, if you look at it, the project is just completed. So overall, there were 17 endogamous groups. We also obtained 325 isolates, and we used metagenomics, and metagenome is again, this is a technical term, but we looked at the total genome sequence of gut bacteria in these 3,400 individuals, and what we come across was that bioinformatically we assembled their genomes, and we could assemble something like 50,000 genomes, and in the genomes and in metagenome we look for the antibiotic resistance genes. So what we found is that for multiple categories of antibiotics and multiple taxonomic groups of bacteria, there is a significant presence of antibiotic resistance. So you take all the classes of antibiotics and all different taxa, there is a presence of antibiotic resistance, which means that even in the gut, and these population includes some of the tribals also. So in all these individuals, different types of organisms carry antibiotic resistance. So how it has come to them, of course, some of them would have had antibiotics also, but some of the tribals did not have access to antibiotics. So, but the antibiotic resistance is present everywhere, and this shows the distribution of the genes across different phyla. So left hand side you will see the overall presence of antibiotic resistance across different phyla, and on the right hand side you will see the mean antibiotic resistance across different phyla. So the point which I wanted to make here is that even our gut bacteria would naturally carry antibiotic resistance genes, and so these people obviously they did not have any infections, no infection from, when the sample was collected, they did not have any infection from antibiotic resistant organism. This is their natural gut microbiome. We had another project which was, again, I left NCCS, was taken over by Dheeraj Dhotre, where we looked at the antibiotic resistant organisms, in sewage treatment plants. This was Gates Foundation funded project. So overall plan was in Mumbai, different sewage treatment plants, we collected samples before, during and after the waste treatment, and we looked at the presence of antibiotic resistance genes. So the results were not surprising across the different, this was just the taxonomic picture of those samples at different locations, but what we found was that this is a little complicated figure where it looks at, it analyzes the metagenome data at different levels, finds out whether the organism is has a resistance genes or not. If it is not found, then it deletes that. If it is found, then it goes next. So it's a complicated analysis, but what it shows is that it calculates the threat for the presence of antibiotic resistant genes and their spread. And if you see in the right hand corner, there is a point dot 1, which means that there is no threat. It is a safe situation. However, none of the samples lie any close to that. All the samples you will find that they lie towards the right hand side, which means that each and every sample that was collected at different time points, always there was a threat of rampant spread of antibiotic resistance through sewage also. So there are multiple reasons for that, because these sewage lines contain some of the hospitals in the city. We also did studies in the sewage treatment plants of those hospitals. So this is again a very alarming study, and again, it again shows the same thing, that these antibiotic resistant genes are, multiple antibiotic resistant genes are spread across different taxa. So it is not that they are present only in one particular group of organism, they are present all across the taxa. So which is indicates a really alarming situation. And there was, this is a mute point, there was a seasonal variation. We studied the beta-lactam group of antibiotics in more details, because the resistance was more for beta-lactam group, and we found that all across the sites there was a seasonal variation in the abundance of different antibiotic resistant genes, and this variation was different in different locations. Again, we really don't know the reason for these differences, but this is something that we observed. And then finally coming to what we are doing at present. So presently at SCAN also we are involved in research related to antibiotic resistance, and we use this particular technique for the identification of microbial pathways and essential functions. So this is basically a large scale mutagenesis study where you carry out mutagenesis in such a way that there will be at least 3 hits in a single gene, and then you do the sequencing, and then you find out the essential genes for either for the resistance of the antibiotic, in this case resistance of the antibiotic, or the mechanism of action for the antibiotic. So there are many variations in the technique, and this is something which we are in one way you lose the essential gene, so you just know that this gene is essential, but the mechanism of action you don't come to know. So there is another variation of the technique where you insert the transposon, but then you also have a promoter, so that a part of the gene is transcribed and you come to know the actual function of the gene. So we are using both these to understand for the discovery of novel antibiotics and understanding the mechanism of action of existing antibiotics. And just to give you a flair for this is not the work we have done. This is a work which is in collaboration with Quadrant Institute Biosciences in UK. So this is a work done by Quadrant Institute researcher, where the simple triclosan, which is ingredient of the toothpaste, they have found out some novel genes in the antibiotic resistance of triclosan and the mechanism of action of triclosan. They have found out novel genes which were not known earlier. So that is one example where to see how this technique can be used powerfully. And same way, this technique was also used in another study for identification of many new secondary resistant genes in colistin resistance in Klebsiella pneumoniae. So conventionally we know one target for colistin, but this study identified this particular gene, DEN-A, which is a novel gene responsible for development of resistance for colistin in Klebsiella, and it was first identified by mutagenesis in this study, but then subsequently its importance was proved by gene knockout, complementation and everything. Its importance was shown. So these are the novel techniques which one can use to find out the mechanism of action of a novel drug, or molecular mechanisms behind development of resistance. So there are, this is again talked about in the morning, so I will not spend much time on that. The strategies to combat resistance, and considering this importance, there is a global action plan that was proposed in 2015, followed by national level plan for India in 2017. A lot of discussions took place while developing this national level response, and there are many priorities of national level response: awareness and understanding, knowledge and evidence, infection prevention and control. So it's a kind of holistic approach which takes care of all the aspects: optimal use of antibiotics, innovation and R&D, leadership. So it takes care of being a holistic approach. It takes care of all the aspects of mitigating AMR threat. And today also we talk about one health approach, where we don't look at infections and environment, animal infection, human infection and environment separately. We consider everything together, and there is a move to mitigate antibiotic resistance using this One Health approach. And in the One Health approach, I mean, improvement of health so that improve the immunity is also one aspect of One Health approach. And as a part of this National Action Plan, Department of Biotechnology established this AMR repository at NCCS in 2018, and it collaborates with WHO, many Indian networks for the which are involved in AMR screening across the country, which includes Maharashtra State Network, Kerala State Network, Delhi Network. So through all these regional networks, and also Armed Forces Medical Services, FSSSAI. So from this variety of sources, the available antibiotic resistant organisms which are circulating in the population are collected in this repository. And today the repository has something like 1,259 well characterized antibiotic resistant organisms. For around 1,000 of these, the complete AMR pattern has been done, and they are spread across 39 genera and 105 bacterial species. And for 70 of these organisms, the complete genome sequence is also known and published. So, like this, there are many resources available in the country for combating the antimicrobial resistance, and there is not a single approach that is going to be successful. So one has to try a variety of approaches, alternate means of therapy. As I said, that infection control is one of the major way by which indiscriminate use of antibiotics is controlled. So if the infections are less, naturally there will be no need for the antibiotics. And a proper antibiotic policy, if all these are in place, then we would be in a position to successfully combat the increasing threat of AMR. So with that, I'll stop here. Thank you very much for your patience.
Thank you so much, sir, for mesmerizing us with the mechanism of resistance. And I think we can take the leads, like many parameters were new to us, like the TRADIS, the mechanistic action, wherein we can find out the mechanism of resistance. So you can take out, because see, we have supportive literature for homeopathic medicines. So if our medicines can also be tested by this method, TRADIS, that will be great. I think then we will not need this preclinical data is well established, which will help us in doing the clinical trials further. So I think we'll take the help of sir. And we have any questions from the audience?
Sure.
Thank you, sir. As expected, the talk was very enlightening. Sir, I wanted to know, what is the role of probiotics? Like, are we overdoing it? Are they recommended, not recommended? Are they adding on to the problem, or is solving it? Thank you.
Thank you. Thank you for that question. It's a very difficult question to answer, because probiotic is a kind of grey area. Grey area in the sense that, unfortunately, the probiotics that are available in the market are all of Asian or European origin. So, how far they are really beneficial in a long term for human health is something which is questionable. And also about whether the probiotic organism should have antibiotic resistance markers or not is an open-ended question. It has not yet been answered. But if you look at the other side, the best example is Clostridium difficile idea, where it is not exactly a probiotic, but FMD has been successfully used to treat the patients with this diarrhea. And there are some smaller studies in India also, where in neonatal infections, people have used commercially available probiotic infections with certain degree of success. So I would say in extreme condition, you are not worried about probiotic being effective and having health benefits, but you just want to control the infection. In such cases, those probiotics have been useful.
But they are commercially available ones are different from what the doctors are recommending.
No, all those are commercially available. I mean, today, what doctors recommend are the commercially available preparations which have been originated from the global strains, not indigenous strains. Now, there are some smaller companies that have come up with, they claim that they have some indigenous isolates, but again, it has not gone through proper scientific evidence to prove the effect. Yeah. Thank you.
So we will move further now. Thank you so much, sir, for an enlightening speech. I would now like to call upon our next esteemed speaker, Dr. Sonal Saxena. She is the Director, Professor and Head Department of Microbiology, Maulana Azad Medical College. Ma'am has been working on AMR since last 26 years, and she is the coordinator for Delhi Network for Surveillance of Antimicrobial Resistance, State Nodal Officer for AMR Surveillance. She is the principal investigator at the state-level Viral Research Disease Laboratory, ICMR DHR, and she is the lab coordinator for the Delhi's COVID-19 Sero Survey. Dr. Saxena has 93 publications to her credit and has been honored with the distinguished awards, including the WHO Fellowship on Antimicrobial Resistance at ICHAR Karolinska Institute, Stockholm, Sweden. Dr. Saxena has also been conferred with FAMER, Foundation for Advancement of International Medical Education and Research Fellowship in 2023. We are honored to have you, ma'am, with us today and invite you to enlighten us on antimicrobial stewardship.
much for having me here, and my apologies for this small glitch. This is what happens when a non-tech-savvy person like me tries to be very tech-savvy. So, you know what is the advantage of being the last speaker? Your word has been made simple by all the speakers before you. So you just have to breeze through a walk on the beach. I'm going to talk a little bit about antimicrobial stewardship, and to enlighten you further, let me take you back many, many years ago when you have any idea how penicillin came into being? Anybody any idea how penicillin was marketed, how it was sold? The molecule was discovered by Sir Alexander Fleming in 1928, but it saw the market in 1940. How and why is a very interesting story. Jane and Florey allowed the same molecule to be purified and made into a compound, and they were ready with the compound in the 30s. But it came into the being because it had to save lives during the World War II. And had it not been penicillin, a million more lives would have been lost due to this. And there were two things which were happening in war. The soldiers were lonely. So the whole of Europe was under siege, and the soldiers were very lonely, and they were getting sexually transmitted diseases. You can now put two and two together as to why that was happening, because they were lonely. And they needed a cure for it. So penicillin initially came as a cure for gonorrhea and not for war wounds as we all think. That it came into World War II, so it must be for war wounds. But at the same time, because it was the first antibiotic to be licensed, it worked like a miracle on the war wounds. Even now, most of the wounds which happen even in the hospital do respond to a certain extent to congeners or further developers of penicillins, as we call it. So everybody thought we have a Superman, you know, those were the comic days, and everybody thought we have now Marvel Man and Superman, and now we can conquer the microbes. And like Sir very wonderfully made my job easier, I always like to believe we are the invaders on a planet which was inhabited by microbes. देखो, किसकी सीट होती है पहली बस में? जो पहले जाके बैठ जाता है या जो रुमाल रख देता है? तो सबसे पहला रुमाल धरती में किसने रखा था? माइक्रोब्स ने. तो फिर हमारी तो आई नहीं, हमने तो उसको उठा के बैठने की कोशिश की है. एंड इस कोशिश में अगर वो कहता है नीचे मेरी सीट है, तो क्या बुरा करता है? आप कितनी भी एंटीबायोटिक बना लीजिए, like sir said, before the antibiotic is made, he is resistant to it. So जो archived strains थे उसमें भी methicillin resistance Staph aureus मिल रहा था before penicillin was marketed. So इसीलिए तो इतने साल लगे उसको बना के मार्केट में लाने में. तो कभी भी कोई सुपर एंटीबायोटिक हो ही नहीं सकती. आप धरती छोड़ दीजिए, that is what they want. ये मेरी बस खाली करिए. And Alexander Fleming ने 1945 में अपनी Nobel Prize speech में कहा था कि the microbes are educated to resist penicillin. At that time he never gave this word of antibiotics, so he said कि जितनी जल्दी आप इसको डेवलप करते जाओगे उतनी जल्दी वो भी रेजिस्टेंट होता जाएगा. एंड नाउ पेनिसिलिन इज रिस्पॉन्सिबल. एंड imagine something which he said at that time, that the man who finally succumbs to infection with the penicillin resistance organism, I hope this level can be averted. वो अगर कवर में है तो पलट रहा होगा पिछले 25-30 साल से कि भाई ये क्या कर दिया इन्होंने. अच्छा, बहुत सारे organisms में ना innate resistance होता है, which even the best of the clinicians don't recognize. So when we go and buy from pharmacy the drugs, we don't realize this, that certain organisms are born in such a manner that they will not, respond to those drugs. How many of you have eaten raw fish? If this was a Japanese gathering, I would have had all hands up. तो नहीं खाते ना. वैसे सिमिलरली ये बैक्टीरिया भी बहुत सारे एंटीबायोटिक्स को नहीं रिस्पॉन्ड करता. एंड वहीं हमारी क्लासिफिकेशन अलग होनी लगती है. तो जैसे in homeopathy, somebody was telling me in the morning that you go by the symptom and you go by the, as far as I believe, you go by the psyche of the person. कि okay, you like salty or you like sweet, or are you tall? Are you able to digest proteins better? Or you like carbs better? Even that makes a difference. That is the precision medicine. We are also gearing towards it with NGS and genomic sequencing and all that. But this is what happens with bacteria. They don't like certain foods. Tetracyclines for Pseudomonas, ampicillin for Klebsiella. They don't open their gates to them. Very simple. These antibiotics cannot enter these bacteria. If they can't, or even if they enter, they are shown the door immediately. Acha to aa ya na nikal ja. So there are these two mechanisms only which work when we talk of intrinsic resistance or innate resistance. The other is an acquired resistance, which I get. If I go to Japan and I like sushi, I will start looking for sushi restaurants in Gurgaon or Delhi or Noida. कि यार मुझे sushi बड़ी अच्छी लगी थी, वहाँ पे मुझे खानी है. Don't all of us here have not. How many of you have been to Italy and only eaten pizza there? But all of you have eaten pizza because it is now freely available. So similarly, they develop resistance to it. I am resistant to pizza. I can't digest it. I can't eat it. I will throw up if I eat it. So I've developed resistance to it. I've eaten so much of pizza probably that I've given up. So a lot of that resistance actually happens by overexposure. You might say, "Ab nahi main kha sakta yaar ye cheez." It's playing havoc with my blood pressure. Or I could develop something by which I have replaced it with something else instead of. आजकल इतने बड़े सारे YouTube वीडियो चलते हैं which talk about healthy mayonnaise, पनीर पीस लिया, सोया पीस लिया, है ना? बड़े सारे चलते हैं. I really sometimes wonder should I waste my ingredients on it or not. So what are they doing? They are replacing it with something else. So similarly, they also replace the target with something else. अगर वो पेनिसिलिन में जाके पेनिसिलिन बाइंडिंग प्रोटीन पे बाइंड करना है, वो उसी को बदल देता है टारगेट को. Now you can't sit on my chair. You want to remember the enzyme theory ना, कि वो इस स्ट्रक्चर का है और उसका रिसेप्टर ऐसा है. अब उसने अपना स्ट्रक्चर ही ऐसे कर लिया तो वो कैसे जाएगा ना? So it alters the target. The simplest version is I don't want to eat pizza. I will not open to the door of Domino's. I will delete Zomato from my mobile. That I will not order. Whenever my daughter goes into that dieting drive, she does this. Swiggy delete कर दिया, Zomato delete कर दिया. Mobile ही तो मैं order ही नहीं. So the same way the bacteria decides कि I will not allow the drug to enter. If I allow the drug to enter, I will show him the door कि तू घुस तो गया, मैं तेरे को use नहीं करूँगा. Or they use a different kind of a metabolic pathway. They bypass the usual metabolic pathway. So these are the usual mechanisms by which the organisms develop resistance. And why they've become trained to it. How? By inappropriate use. By a use which is, you go to a pharmacy, you never finish your course, you take 3 days of treatment instead of 14 days of treatment. So inappropriate is very good. Lack of infection control. कोविड नहीं आता तो हाथ धोना ही नहीं सीखता भारत. Guaranteed. स्वच्छ भारत अभियान में मोदी जी कह के थक गए, भाई हाथ धो, स्वच्छता रखो. जैसे कोविड आया, everybody was washing hands 1000 times a day. इतना ज़्यादा कि माइक्रोबियल फ्लोरा ही गायब हो गया हाथों से. Then these influx of patients, once the patient, the kinds of patients we get in Maulana the Lok Nayak hospital that we have an OPD of about running into 10 to 15,000 per day. Because we have an IPD of about 2,500. So the kind of patient that we get is already been to 10 places and then landed in Lok Nayak. तो उसको 6-7 antibiotic तो मिल चुकी है. So his flora, like sir was saying, is completely resistant because now he's colonized with that resistant flora. वही बचा है ना जो resistant है, sensitive तो मर गया इतनी दवाई खा के. Then the waste management protocol. The biomedical waste management plays a huge role when we compare to this. There are multiple studies which talk about the residues of antibiotics in sewage, and they destroy our environmental flora, which plays such an important role in scavenging the atmosphere and creating the environment health. And the most misuse happens is here. How many of you take an antibiotic for URI? Honestly, completely, I am not judging you. Very honestly, जुकाम हुआ, खांसी हुई, आपने एंटीबायोटिक खा ली. Almost everyone has taken. चाहे आप मानो, चाहे ना मानो. एजिथ्रोमाइसिन खाई थी? चलिए, maybe because you are homeopathy doctors, that is why. That's why. Because you had an alternative. Yes. So what happens is that there is a grandma adage. If you take जुकाम की दवाई खाओगे तो 7 दिन में ठीक होगे, नहीं खाओगे तो 1 हफ्ते में ठीक होगे, है ना? But we don't sit on it. Another common illness where it is misused is diarrhea. आज आपने बड़ा heavy dinner किया. कल मेरे in-laws की anniversary थी. We had a huge, they are from Lucknow, so we always their anniversary celebrated with a Lucknowi meal. The shami kebabs and the khamiri rotis and all that. And in the morning, I see everyone in my house popping a antacid. Now what happens is that when you pop that antacid, a very simple thing happens. It changes the pH of your stomach. The moment it changes the pH of your stomach, whatever you eat today is going to be very risky. Because acid is one of the biggest inhibitor of bacteria. So when the acid is reduced, even if you take a very low dose, say 10 to the power 2 instead of 10 to the power 5, it is capable of causing infection. तो अगले दिन डायरिया हो गया. फिर हमने बोला यार परसों खाना खाया था ना, ये बहुत बेकार रेस्टोरेंट, इससे खाना मत मंगाना, इससे डायरिया होगा. डायरिया तो आपने खुद किया. Your body acid would have taken care of that much of bacteria which went inside, but you never allowed it by suppressing it. So these are just 1 or 2 examples, And then the moment you have diarrhea, people will again pop in an antibiotic. Okay, I want to खा लेते हैं क्योंकि diarrhea हो गया. It could be food related, it could be a food allergen, it could be just a gastric hurrying because you've eaten too much of spice, oil, which your body is not used to. So this kind of thing, people who are on perennial dieting, if they eat even a little bit of spice, of oil, of fat, they will have some amount of maybe loose motion or frequency of stools will increase and they will think infection. So that doesn't happen. So just wait. They will not work in your cough and cold anyway. And this antibiotic pressure is ultimately going to select out, because when you eat these antibiotics or when you give to others, the resistant flora will only perish, will only survive. The rest will all perish. And the susceptible one will then start becoming resistant because there is a gene transfer which is happening at a horizontal level. Bacteria all divide by binary fission. So when they divide by binary fission, they take extra genetic elements as well sometimes. That extra genetic element is going to transfer one mutant gene of antibiotic resistant to another, and then they are dividing by binary fission, so they will go on giving it to their progeny. And this concept of one health that Sir was talking about, that everything is interlinked. You use antibiotics in animal feed, you use antibiotics on plants, on animals, everything goes into your environment, into your sewage. The further exposure to antibiotic happens through food, through drinking water, and all these things happen. Colistin a little while ago was used one of the biggest poultry growth promoter. You add antibiotics to the poultry feed, and what happens is that because they will not get infection, they give you a very good yield, and they allow the accumulation of water in the muscles, which allow the poultry to become heavier, because ultimately it is sold by weight. So that is the reason why it was used. Now, although there are many laws and it is bad, but I always believe it is the human health which is responsible for the state that we are in. And it is my house on fire and I have to rectify it. I cannot say कि animal वाले करेंगे, तभी मैं जगूंगी, तभी मैं कुछ काम करूंगी. And the original superbug was probably stabbed because it was the first drug to be treated with penicillin, and the first superbug was probably methicillin resistant staph aureus when it developed resistance to methicillin. And then came various foodborne bugs like E. coli and Salmonella, which started becoming resistant. And now we have Salmonella, which is resistant to even the last resort of drug. When we started the DENSAR, as we call it, the Delhi State Network for Surveillance of Antimicrobial Resistance, in 2019, we were not getting any safe trials on resistant Salmonella. And now, from these 24 labs that are under us, and now we get approximately 1 to 3% in the past 2 years. And I'm sure when I compile 2024 data, this will be in 2 figures. And then came the sexually transmitted infections like gonorrhea and chlamydia, which started becoming resistant. And then the hospital pathogens, Clostridium difficile, Acinetobacter, Enterococci, extremely prevalent in hospital. They love to survive on those stainless steel surfaces which are there in the hospital, and they become multi-drug resistant. Then of course the global threat of tuberculosis, or maybe the threat was there, we've caught it now because of the availability of detection. So all these have actually acted as an eye-opener, and people have started thinking that we have nothing. Why is it happening is because the pipeline is getting drier and drier. It takes 20 years to get a molecule, and there is no molecule right now which is into that 20-year clinical trial phase. They're all variations or additions. Ceftazidime में तो avibactam add कर दो, meropenem में तो vicorbactam add कर दो, that kind of things. So it's kind of a superbug story in which the bacteria will travel from here to there, and the best part was that if you look at the global travel, we all thought COVID is not going to happen. It's not going to leave China because China is so strict. It will not leave China. 19th December, when the first news came of the patient dying of COVID, we all thought it's not going to come. And within 1.5 months, it was knocking at our doors and we had nothing to stop it. And this was when the whole world was on alert. If you remember MERS-CoV, I don't know, and the original SARS in 2008, the patient traveled from Australia to Canada, and within a span of 48 hours, the disease, he had spread it to all the people in his aeroplane. And the disease had spread from one corner of the globe to another in a matter of 2-3 days. So the antibiotic deployment gun has been silent for many years now. It's been going on intermittently, but now it is almost silent. We've not had a new antibiotic after 2004. Whatever we have are the congeners to this. We add something to another to make it more. And we are nearing a post-antibiotic era where people will die of a very, very small infection. Choti si keel lagi, wound infection hua, and it will not respond to any antibiotics. So whatever big the problem fire is, they all start small. And we have to catch it when they are small. Otherwise, and this is already covered, so I will not talk about it. So what will happen is that the duration of illness will be much longer. You can treat typhoid now in 7 days, you might require a therapy in 30 days, you will increase healthcare associated infections, you will increase mortality, you will have to treat with more expensive drugs. Even now, the newer molecules which are coming from US, their cost of therapy is approximately 15 to 25,000 per day. And if interleukins come into being, it will be 1 lakh per day. And of course, increased burden on the healthcare system, because in our healthcare system, in India particularly, majority of the healthcare system is paid by the state. It's not paid by the user. So if it is paid by the state, it is a burden on the exchequer or in the board on the state. State kahan se nikalega? Aap hi se wapas nikalega. So the patients will become reservoir of these resistant and will create a longer duration and in the environment. So is it the end of the road? Not yet. We're not yet locked up in the jail and not yet started responding. We need to lock up the microbes. So the solution could be that we start making new drugs faster and sooner, like we did with COVID vaccines. Allow them as research use only and bring them out. It's again a very, very challenging and not a very lucrative thing to pharma companies. Why? Any idea? Why making new antibiotics is not a lucrative thing to many pharma companies? What do you think could be happening? People who have not taken antibiotics ever. Why is it not very? If I was owning a pharma company, I would not invest my money into making an antibiotic. It's a self-dying molecule. Tomorrow the bacteria will become resistant to it and it will lose its market. So I spend my money, my my time, my research on a molecule which has got a very limited shelf life. Within few years, everything will become resistant and it will lose its potency. So we can reduce cross infections, have a very severe infection control practices, and the brilliant success we have seen in COVID. We all wore those space suits and still got sick and landed up at home and were quarantined for 14 days. Remember? Then 7 days, then 5 days, then 3 days. Then in my department fever उतरते ही आ जाना, क्योंकि lab में 6 sample आ रहे हैं. So it's not a very practical solution. Can we preserve the efficacy of these? Yes, that is the only thing which is going to work. Can we preserve the efficacy? Whatever is there, let's use it with a lot of resource. अगर घर में थोड़ा सा आटा बचता है तो माँ कहती है यार आधी आधी रोटी खा लो, पर खा लो, है ना, सबका पेट थोड़ा थोड़ा भर जाए. So we need to optimize the therapy without losing life and without losing. So that is where the antimicrobial stewardship comes in. And आपने कितने प्लेन क्रैश होते सुने हैं अपने लाइफ टाइम में? एक या दो, है ना? So I'm over 50. I've heard maybe 3. Those in the 30s and 40s, a relatively younger crowd here would have heard about 1 or 2. एक वो मलेशिया वाला गायब हुआ था, एक आध. क्यों नहीं होता ये? Why doesn't it happen? The airline industry functions on a steward mode. एक को चेक करने के लिए दूसरा होता है, दूसरे का चेक करने के लिए तीसरा होता है, तीसरे का चेक करने के लिए चौथा होता है. वहाँ कोई भी नट बोल्ट एक पे नहीं छोड़ा जाता. आपने देखा है मोमेंट प्लेन लैंड्स 3-4 इंजीनियर चढ़ जाते हैं उसके अंदर. वो पायलट उतर जाता है जो काम कर अब तक आया है, और वो इंजीनियर पूरा प्लेन टेक ओवर कर लेते हैं. कोई नीचे से कस रहा होता है, कोई ऊपर से कुछ कर रहा होता है, क्वालिटी चेक. फिर 2 of them will come out, another 2 will go. So, and then if something happens, if the plane crashes, how do we prevent further crashes? By a black box which has recorded everything, what has happened. So we need to have something which monitors, you know. कोई भी आदमी रेड लाइट बंद नहीं करता अगर वो वाइट यूनिफॉर्म पुलिसमैन को खड़ा देख लेता है उस रेड लाइट पे. दूर से चिल्लाने लगता है वो खड़ा है भाई धीरे कर ले, है ना. So we all work very differently. This is called a Hawthorne effect. We all work very differently when somebody is observing. So stewardship is that we have entrusted the responsibility very carefully into someone's hand. एक एयरलाइन में स्टीवर्ड होता है, होटल में स्टीवर्ड. अगेन होटल इंडस्ट्री आल्सो वर्क्स लाइक दिस. वो एक कमरा साफ करके जाएगा, फिर दूसरा देखेगा, फिर तीसरा एक फ्लोर मैनेजर है जो आपको पूछेगा कि सब ठीक है ना मैम. So similarly, antibiotic stewardship is also a coherent set of actions which promote responsible use because the actions are getting observed and the further actions are limited to that observation. So it's an activity where I give an appropriate dose, I give an appropriate duration, and I give the right drug. So it is defined as the right drug, at the right time, with the right dose, by the right route to the right patient. And it should result in the best clinical outcome. ऐसा नहीं कि मैंने एंटीबायोटिक दी और वो मर गया. Antibiotic stewardship is not about not giving the antibiotic, but giving the antibiotic to those who need it and withholding when it is not needed. And it should not only have the patient best outcomes, it should also, work to decrease the resistance. So, how do we decide the drug? How do we decide the right drug? If the patient is actually sick and you think that it needs antibiotic, for example, pneumonia patient, sepsis patient, they will need antibiotic. Urinary tract infections are all caused by bacteria. So if it is a proven urinary tract infection, it will require an antibiotic. Now if the antibiotic is needed, say azithromycin comes in 2 dosages, 250 and 500. Now to a person who is weighing more than 60 kgs, if you give 250, it is not going to work. कई बार आपने देखा मैं आधी गोली ले लेता हूँ. अरे भाई, आधी गोली is not going to reach that level. See what happens is the antibiotic has to be achieved in your blood or in your serum. It has to reach a certain level to kill all the bacteria below it. अगर वो पहुँचेगा इतना तो ये वाले तो सारे बच गए. Right time. Right time means if the person is sick and I feel that whether I should wait for the results from microbiology to know, or I can't wait. Meningitis patient, I have to start the antibiotic immediately. I will not wait for lab results. So I will take out the sample and give the antibiotic and then wait for the result to change it. But if it is a patient maybe of UTI, I can always tell them, ek din aur ruk jao, and tomorrow we'll have some growth or something, then we'll start the antibiotic. Of course, the dose and the route we all understand. Then the purpose is to optimize patient safety. Where is the safety coming into being? Because the antibiotic, anything which is given in suboptimal doses, is sufficient to kill the normal flora because they are pretty sensitive population, but it will not kill your pathogen. And it has to achieve a certain concentration in the blood to kill the pathogen. Improve clinical outcomes. We all work only for one thing in life, that is professionally, is to improve the clinical outcomes. अगर आप कितनी भी study करते हो, research करते हो, paper छापते हो, your ultimate aim is this study beneficial to somebody in future in improving the outcomes? Of course, control the cost. Reduce resistance, Clostridium difficile infections or any other toxicity, and prevent unintended consequences. And if you realize in this diagram, everything is going up and down. That means it's not just one thing which is in silo. They are all working together. So if you run a stewardship program in the hospital, it needs to have a push from the leader. Nothing, especially in India, nothing happens unless there is a push from the leader. मोदी जी बीच में बोल देते हैं कि हमें टीबी हटाना है 2030 तक. तो suddenly सबको याद आ जाता है. World Antibiotic Awareness Week होता है. Everybody remembers that we are all geared up for this purpose. And then every program should have an accountability. You run a teaching program, you always appoint a batch in charge. कि ये कौन चलाएगा? Who is accountable? Why? Who is running it? What are the various actions that we take? What is the pharma expertise that we have? Then how do you track the antibiotic use and antibiotic resistance? I'll come to these in a little while. What do you report and how do you educate? And this is also an educational activity that we are all having today. The theme this year is educate and advocate. So the education is always ongoing. आपने clinician को बोला भाई ये antibiotic क्यों नहीं होगा? Why are you asking me? आपको उसको पता ही नहीं है antibiotic resistance के बारे में कि इससे कुछ हो सकता है. And not only that, the patient education is also very important. Very often our private practitioners, those doctors who are in practice will come back to you and say, "Ma'am, they come, वो कहते हैं डॉक्टर बेकार है, उसको लिखा ही नहीं, vitamin लिख के भेज दिए." And this happens to you also. When you go to a doctor, you expect him to prescribe you something nice and expensive, as if you walked into a Ritu Kumar's couture where you want a dress. अरे भाई जितने बीमार हो उतनी तो दवा मिलेगी. But that's how people are. ये पैसे दिए हैं डॉक्टर ने 1500 रुपए, कुछ दिया ही नहीं इसने, 1500 रुपए भी ले लिए, है ना? So the choice of antimicrobials to prescribe, there are various strategies. We can use education strategy, like I said. We can use antibiotic cycling strategies. Recently, we've seen a very unusual thing. We have seen that the sensitivity to tetracycline and cotrimoxazole is coming back because clinicians stopped using it. It's a cheap antibiotic. Suddenly, organisms were resistant to it for many years. So now the sensitivity is. So this is called antibiotic cycling. You can use levoflox for some time in your formulary and then switch over with something else. Formulary restriction strategies and computer decision making. Formulary restriction, I'll come to that. And of course, review and feedback strategies. So the 5 R's is that we reduce, we refine, we replace, we review, and we responsible. So what do we reduce? We reduce the unintended use. Those who need it, those who don't need it. What do we refine? We refine using a narrow spectrum agent rather than a broad spectrum. Broad spectrum is going to kill all the antibiotics. Broad spectrum is like a missile which Beirut and Israel and they keep doing every day. वो देखता नहीं है इंसान है कि जानवर है मवेशी है मार देता है. But when you have a narrow spectrum, then it is intended only for the selected. So only the gram-positive bacteria will be killed, or only the gram-negative, or only this particular antibiotic organism will be killed. And we replace it with a lesser toxic and the broad to narrow spectrum. we review. 48 hours, I always tell our clinicians, please review your prescription. Does your patient need antibiotic? Now you have a report. Do you need to change it to a narrow spectrum? Do you need to switch from an IV to an oral? If your patient is eating food, it can also swallow a tablet. So remove your cannula because it is acting as a site of infection again. Something inserted into your body, it can get infected. And who is responsible? Unless and until you make somebody responsible, everybody will say, पता नहीं उसने देखा होगा मैंने क्या दिया, मैंने नहीं देखा. And there is a 30% rule. 30% of the antimicrobials which are given in the hospital are inappropriate. And almost 30% of the hospitals of the antibiotic prescriptions are not required. 30% of the surgical prophylaxis are not required. Only a single dose is required. Delivery हो रही है, antibiotic दे दिया. क्यों भाई? It's a normal procedure. घर पे होता था पहले. Antimicrobial cost, they account for 30% of the cost, and if you initiate the antibiotic stewardship program, you save approximately 30% of the cost. And the microbiology guided therapy is very, very important because it allows you to review your antibiotic, to reduce your dose, to switch from a broad spectrum to a narrow spectrum, make the decisions of going from an IV to an oral switch. Have you seen this? Youngsters must have seen. It's my favorite because it reminds me so much of the microbiology that we do. What does he say? His favorite word is patients will always lie. Patient will always lie. You cannot get any amount of. You ask an STD patient, did you go outside your marriage in HIV clinic? He says, oh, madam. तो ये कैसे हुआ? Blood लगा? ट्रांसफ्यूजन एक्सीडेंट हुआ था? नहीं मैडम. इंजेक्शन लगवाया किसी को एक्स से? नहीं मैडम. And then the counselor will look at us and say, क्या लिखूं फॉर्म में? So patients will always lie, either out of embarrassment or out of not exactly embarrassment, maybe out of sheer guilt sometimes. So you have to decide. आपने antibiotic खाई थी, कोई लंबी गोली, छोटी गोली तो नहीं दी थी. We are trained to ask the patient. We all are, you all must be. छोटी गोली, बड़ी गोली तो नहीं दी थी. मैडम मैं तो गया ही नहीं कुछ. वो बुखार की गोली दी थी. कैसी थी? लाल और पीली थी. And the moment it says लाल और पीली थी, you know it's a capsule. So it must be an antibiotic. No antipyretic is sold in capsule. It is meant to dissolve in the stomach acid. So it has to be a tablet. So they will always tell you, nahi hua. So whenever why diagnostic stewardship becomes important is for these kinds of patients. Because you will believe that this is the first sample that I am sending, and then nothing is growing in the culture because the patient has lied to you about taking antibiotic. So why does diagnostic? You evaluate the patient, you do a test, and you give empirical, and you say कि ये report आएगी फिर हम change करेंगे. ये बहुत ज़रूरी होता है. I always tell my students, उनको ये बोल देना अगली बार जब change करेंगे तो मैं फीस नहीं लूँगा, वरना वो लौट के नहीं आएंगे कि तू फिर से फीस ले लेगा. तो you say कि 3 दिन बाद ना फीस नहीं लगती है, फीस 5 दिन बाद लगेगी, फिर वो शांति से आ जाएगा 5 बार तेरे को रिपोर्ट दिखाने. So the diagnostic stewardship has to go hand in hand with antimicrobial stewardship. It cannot go in silos. So when you are ordering the test, what sample? Do we have the information on this form? Do we know whether a patient has taken antibiotic? Collection कैसे हुआ? Sample ठीक से आया कि नहीं आया? Fridge में तो नहीं रख दिया? Reporting ठीक से हुई कि नहीं? All these factors will also determine the success of your antimicrobial stewardship. So, and then there are a lot of point of care tests. There are a lot of tests which can actually give you an idea about whether this is a bacterial infection and you should start antibiotic, or is it just a viral infection and you do not need an antibiotic. Okay, I'll skip these. So when you initiate an antibiotic therapy, we first thing that we look at is what is the probable site of infection. Think in your mind that what is a probable site of infection. Consider what is colonization and what is infection. Colonization is mere presence which is there without any symptom. And if what are the rates? अगर ये staph aureus आ रहा है और 60% पेनिसिलिन को रेजिस्टेंट है तो पेनिसिलिन देने से एम्पिसिलिन देने से कुछ नहीं होगा. The rates of most of the gram-positive organisms nowadays are nearing almost 100% to ampicillin. तो ampicillin तो आप placebo effect के लिए दे रहे हो फिर. So you have to monitor the patient very, very carefully when you are following these protocols, and we'll go to these if there is a, what will happen if there is an antimicrobial stewardship team. Because if you are making a program, you will have to have a team. So you will have a lot of people in that team. You will have members of that, clinical pharmacist होगा जो dose monitor करेगा, infection control prevention वाला बंदा होगा जो आपकी status देखेगा, microbiologist जो आपको report देगा, epidemiologist who is going to tell you that what kind of pathogen are prevalent in this area and what you should prescribe, and of course your staff and residents which are there. So you can have a front end or a pre-authorization. यानी देने से पहले आप front end stop कर रहे हो, and there is a back end. Black box वाला example था ना, तो black box क्या है? Back end है कि front end है? Back end है. आप बाद में रिव्यू करो प्लेन में हुआ क्या था, तो आप सारे प्रिस्क्रिप्शन निकालते हो और कहते हो यार डॉक्टर सोना बड़ी खराब है, इन्होंने तो 100 में से 90 लोगों को एंटीबायोटिक दे दी और वो दे दी जो देनी नहीं चाहिए थी. तो फिर अब मेरे को बताएगा कौन आके यार हेड ऑफ द डिपार्टमेंट है, मैं तो नहीं जा रहा. तो कम्युनिकेशन इज अनदर बिग इशू व्हेन यू डील विद एंटीबायोटिक स्टूडेंटशिप. आप मुझे आके बोलोगे कि मैडम आपने 100 में से 90 को दे दी, मेरे पास 90 के कारण है, चल अपना रास्ता ना. And this happens. So the person who's responsible for this in a very key position. So you have to develop a feedback mechanism or a computerized mechanism that these many antibiotics you gave which were not required. या आपके यहाँ से कल्चर तो 5 ही गए, एंटीबायोटिक आपने 100 को दे दिए. फ्रंट एंड इज जो स्टीवर्ड और पायलट होता है ना आपकी फ्लाइट में, जो क्रैश होने नहीं दे प्लेन को. That means I have already restricted that you can only prescribe these 4 antibiotics and these 8 you cannot prescribe till the senior most person or a pharmacologist or an infectious disease physician has seen it. When they see it, only then you will be able to prescribe it. So that's restricted, and you can have a combination of both. For emergency, you can probably have a restriction, and back end is for the other part. So a physician will write order antibiotic. This is how the back end and the front end will work. Then there are various supplementary. You make an antibiotic guideline, you make a formulary. We all do that in our listing. But follow up करना तो हमारा काम है. Bacterial pathogen, priority pathogen I will not cover. I will only tell you this, that the carbapenem-resistant Acinetobacter vomini is actually a huge problem. When I look at the data, yesterday only I was compiling for making a report for the first half of 2024, and I realized that we call this carbapenem-resistant Acinetobacter vomini. This crab was taking majority of the pie में ना आधे से ज़्यादा वही था. But unfortunately or fortunately, most of our hospitals are tertiary care centers. Most क्या all. So the data that we get is actually skewed towards sicker patients, those who are requiring more admission and therapy. But this is the state of our hospitals. And one of the person sometime back told me, the lady who monitors the national AMR network, that I am scared to go to hospital in my old age. I'm really scared to go to the hospital. That I don't know if there will be an antibiotic to treat me, which will be left. Then there is an aware classification which makes everybody aware. So this aware is actually a mnemonic. A for access, WA for watch, and RE for reserve. So access is that they are accessible at a primary healthcare center. They are the least toxic antibiotics and they can be given on an OPD OTC basis. Watch is that there is a possibility of development of resistance and I keep it for certain diseases. For example, I keep ceftriaxone for typhoid. Reserve is that they respond, they work when everything else has gone out of use. So I have to be very, very careful, and these are the ones which also form as the restricted part which we dealt with. And this you're all aware of, the red line. Sir also talked about it. The red line, which is the red line campaign, where they try to restrict sale of antibiotics. Many of my meetings on AMR, I've heard people say, why can't India restrict OTC? India can't restrict OTCs because there is no last mile connectivity of a doctor. You know, we all sit in Delhi, Mumbai, and we think that the country is like this. The country is not like this. The country has no doctor in a peripheral health center, no nurse. The Anganwadi and the Ashas manage deliveries there. So in that area, if there is a complete ban on all, even access, it's going to create more health issues probably than this. So there are multiple challenges which happen when you try to. The first and the biggest challenge is that I don't want to change. That is the biggest challenge. You know, when how many of you use UPI or Paytm or GPay or this? Everyone, no? You know, when I started using it last year in May, probably one of the last people, because I was always scared and my salary account So one day I attended a lecture at Labasna, the Lal Bahadur Shastri Administration Institute in Missouri last year, and this year in July, and there we were having a talk on financial literacy for women, and there she said that men don't think like this. Nikal lega to nikal lega, rabbi aasan hai. But the women think that ye mere paise nikal lega. So it's not your batua anymore. And the safest, she said, right now, with the multiple layers of encryption, is actually UPI. The rest are all private banks and they don't have this thing. She was from the finance ministry. And it got me thinking, and sitting in the auditorium like this, I, approved my UPI. So there is a behavioral change which is required sometimes. Sometimes you just need that push. Somebody has to tell me that I cannot prescribe that antibiotic. There will be a few which will be stubborn like a resistant bacteria. And I tell them on face in our hospital that, yaar, sir, aap bahut resistant ho. Bacteria se zyada aap resistant ho. To change. We all are change. You know? When Elon Musk launched his self-driven car, everybody said, ye pakka crash hogi pehle din hi. क्यों भाई? वो जो रॉकेट जाता है उसे कौन चलाता है? वो भी तो अपने आप ही जाता है. वो जब अननोन जगह में जा सकता है कि ये तो गाड़ी है सिर्फ. So we have a habit, human beings have a habit of doing away with the regulations. अगर कोई देख नहीं रहा तो रेड लाइट तोड़ दो. और ये इंडिया में तो बहुत ही ज़्यादा है, बाहर भी है. इंडिया में तो बहुत है कि अगर कोई देख नहीं रहा तो ये काम कर लो. So there is a very disregard for regulations. Whether आपने restrict किया या नहीं किया, मैंने तो दे दिया अपने पेशेंट को. तू बाजार से खरीद ला, ये नहीं देंगे. This also happens sometimes. Then the antibiogram that we tell our clinicians in around February every year, we make antibiogram for our hospital and we tell them that, see, this is your bacterial profile and this is the resistant rate in your hospital. And they come back to us and say, क्या बात कर रहे हो मैडम, इतना resistance थोड़ी है. So they are not even ready to believe that. I am also not ready to believe when I see that on paper कि ये क्या हो गया. And there is a competition, there is a pharma push कि मैंने दवा नहीं दी, like I was telling you, उसने दवा दे दी, उसके पास जाते तो हम एक दिन में ठीक हो जाते हैं. And there is no champion कि ना ये antibiotic AMR champion है. तो ये AMR champion है तो ये हम इसकी बात सुन लें. अगर ऐसा कुछ हो, we don't designate somebody in the hospital. हमें लगता है छोड़े है, छोटा सा काम. And everybody is very busy with the load that I was telling you, 10 to 15,000 OPD per day. Who has the time to ask about antibiotic history, send a sample, counsel the patient कि भाई तेरे को कुछ नहीं होगा, तू 3 दिन रुक जा, अपने आप ठीक हो जाएगा. That will need some counseling and some confidence building with the patient. And there is also a lack of interdepartmental coordination. Very often we used to get this complaint. Our hospital unfortunately doesn't have an information system as yet. So very often we used to have a reporting yati hai. So we started making WhatsApp groups. That we made one WhatsApp group with pediatric ICU, one with radio onc, one with medicine unit, one with surgery ICU. तो हमारे रेजिडेंट सुबह रिपोर्टिंग करते हैं, वो फॉर्म्स को रिपोर्ट्स को व्हाट्सएप पे डाल देते हैं कि आप देख लो. उसमें सिर्फ उनका उधर का ट्रीटिंग डॉक्टर है और इधर की ट्रीटिंग डॉक्टर है. no interface in between. So all these you can do it on your own and we can increase it, but as a common man, be vigilante about antimicrobial resistance. Thank you.
So ma'am, thank you so much for adding humor to the lecture and teaching us about AMR stewardship. But I have a question. Can homeopathy be integrated with the AMR stewardship team, like we were seeing?
Only when you have a mixopathy kind of a hospital, because like our hospital will not have homeopathy. But even if we'll have only one. Whereas we have about 1200 doctors to run the hospital, which is 2000.
So can't the cafeteria approach be implemented? Because now it's a global concern, and because you being the state nodal officer for AMR surveillance, we really want that you open avenues for our battle. Because I mean, the AMR stewardship parameters.
Majority of the hospitals under the central government I know have got now an Ayush clinic with them, where there is a physician sitting there. So I'm sure the clinicians do take their help as and when required. But it's not there in my hospital right now. But we are delivering.
But something documented, something can be documented to make into account.
We can do, see, only evidence works.
That's true.
I always tell my residents that they will not listen to you unless you tell them that you have 70% MRS. Nobody will listen to you. So the same way the evidence works. Have to have some combined studies, generate some evidence, and then maybe we can convince the bureaucrats and other people to have.
Like we are doing that escape study also. I mean, maybe we'll be requiring it because जो आपने बताया कि antimicrobial stewardship में right dose, right medicine. I think we are as a homeopath we have all our laws which go synchronous with the stewardship. So I think we are better placed and we can be a part of this team if you guys take it forward. Thank you so much.
So to ask about the quality of antibiotics also matters for the AMR because sometimes it is said that purchase of this brand or this brand.
And when you are talking of the right dose and the right duration, it is my belief that 500 gram will contain 500 milligram, will contain 500 milligram. If it contains 200 milligram, then it is a suboptimal dose. If I am talking about giving DD and it is the two doses are not even sufficient for a one dose, then of course it is there.
Are there any studies or some work is being done where we can see the resistance is, some microbe is resistant to some antibiotic resistance, some microbe and it is getting converted to sensitiveness with, are there any studies going in the conventional medicines or?
No, I don't think I can enlighten on that because our belief is that we remove the resistance. That's how the allopathy works. The purpose of antibiotic is to not do anything to the body but to kill the bacteria. Now in that process it also kills the good bacteria and also kills the harmful bacteria. Good more and harmful less. Because the good are more in number. So we believe in removing that thing. So sensitive, whether it is becoming sensitive or not, it will not going to lose its gene. No? It's acquired that gene now. Every time it multiplies, it will give the gene to the other one.
But in one of our study which we have done on annular TB, with add-on homeopathy to the standard of care, when we do the cultural sensitivity reports, we used to find that earlier the patient was resistant to INH, rifampicin, ethambutol and other drugs. But when we have given the homeopathic add-on medicine, say for the complete course, for the 2 years, 24 months standard treatment, we found that in some of the cases, the patients who were earlier resistant to some of the drugs have got sensitive. So what does it mean? Even at that time, we are not able to get
What
it.
happens is that we are looking at a population of bacteria. We never look at something which is a single. So if we look at the population of bacteria, there will be some amount of heteroresistance. It will be a mixture of sensitive as well as resistant pathogens. So maybe now the resistant pathogens, maybe with the help of homeopathy, have finally responded, and it is only the sensitive ones which will be taken care of by that particular drug. Because what I believe, homeopathy is more of an immunomodulatory system. So that is why I asked you how many of you took azithromycin. Azithromycin was not given as a drug against the virus. It was given because it's a very good immunomodulator. It works inside the cell and it plays well with your body's innate by your own individuals.
Thank you.
Antimicrobial.
Ma'am, we were talking about colonization of pathogens versus infection of pathogens. So colonization, of course, is asymptomatic and we discover it accidentally when the patient is admitted or something. So is something done to treat colonization as well, or you wait for the symptoms to show up?
Those colonizers, it's like if my house is already full with 50 people, I will not invite a troublesome person to my house. I'll say, "Mera ghar bhara hua hai, mat aaiye." Colonization is that. There are already bacteria which are colonized. They compete with the pathogen for resources. And they will not allow a pathogenic bacteria. So one, they are good. Secondly, if you start eradicating colonization, then you end up in a bigger mess than this. Because you will require huge amount of dosages and they are not going to work. Very often what happens is if there is a resistant bug which is colonizing, it might spread to other people and that is what happens in ICU. So there are certain nasal decolonizers and other things which we do if we take it as an infection control practice. If we find somebody's nasal flora or axilla is giving resistant MRSA, then we decolonize it. There are
These colonies do become trigger for infection, or they get triggered.
They are different type bombs. They can, because the immunity level of your patients is very, very low when they are in ICU. So that is why I said infection control practices are extremely important.
So in fact,
If
that
there
would
is
be
a proper hand hygiene.
yeah, because I think that is one scenario where treating the patients who are already having a lot of colonies in them unknowingly or so. So would be it will become a very big challenge compared to a person.
No, we consciously never try to treat a colonization.
But what I'm saying is that this could probably be a very good scope for homeopathy to come in, because in homeopathy we do not trigger into some symptomatic this
But
thing.
you will go with the symptom. No, there is no symptom of the colonizer.
We can but then what we can see is ultimately that whether with homeopathy the colonies are produced or not, even the colonies that
Yes, in vitro you can check in vitro. In vitro you can, yes, definitely. We did one of the studies with what is that called? Active biocompounds from cucumber, haldi.
Okay.
We did one of these studies and we did one with tamarind, where we found that the tamarind, the haldi extract that we use to inhibit bacteria, you know what amount you will require to kill bacteria? 500 grams of haldi every day, a 60 kg person will have to take.
I'm sure homeopathy will have more encouraging results than that.
So,
Microdose.
that is why the hype of that, turmeric died soon after, because the active compound is so less in it. And the purity cannot be asserted of that compound also. So whatever we were doing this with Delhi University as a combined project, and we had to give it up after pilot because the results were very bad.
Thank you, ma'am.
Thank you so much, ma'am, for enlightening us. I think this was an untouched topic, and you have actually opened avenues for us. You have tickled our brains to take up new projects in this direction now. Now, I would request all the dignitaries to please come up on the dais. DG sir, DDG sir, Dr. Inauguration and in charge. Speaker, Dr. Yogesh sir. I think we had a great learning from these two eminent speakers, and we are really honored to have them with us today to mark this one-day awareness campaign on AMR. I would request our DG sir to kindly felicitate our speakers now. First, Dr. Yogesh S. Sarjit sir. A token of gratitude and for the enlightening lecture. Thank you, sir. felicitate Dr. Sonal Saxena ma'am. Thank you so much, ma'am, for being with us today. We had a great learning experience today. Now I would invite Dr. Renu Mittal, Scientist 4 Admin in charge, to deliver the vote of thanks.
आप सभी को मेरा नमस्कार. Just to sum up about this one-day awareness workshop on AMR, I would like to say that there is a need for traversing the message to judicially use the broad spectrum antibiotics, and we should, as ma'am has mentioned, we should use the narrow spectrum antibiotics, and this message is to be traversed to the primary healthcare center, physicians, the conventional medicine our counterparts, so that we can reduce the antibiotic and antimicrobial resistance first thing. Secondly, as ma'am has said, the lesser use of antibiotics for the post-surgery prophylactics. And here I would like to add, some of the homeopathic medicines have a very good results when we use it in the post-surgical conditions like dental procedures and the postpartum hemorrhage. So, though there is not much of the evidence in that direction, but that can be taken up with integration in integrative models. Thirdly, as Dr. Yogesh has suggested, we can test the homeopathic medicines as antibiotics for the AMR in lab models. And for the last, for the creating awareness, communication and advocacy and enabling behavior change are important for combating the antimicrobial resistance. With these words, I would first like to thank हमारे महानिदेशक महोदय डॉक्टर सुभाष कौशिक जी का मैं आभार व्यक्त करती हूँ जिनके नेतृत्व में हम सभी यहाँ एकत्रित हो पाए. माननीय महानिदेशक जी का मार्गदर्शन और समर्थन हमेशा हमारे लिए प्रेरणादायक रहा है. हमारे उप महानिदेशक श्री डॉक्टर सुनील राम टिके का मैं धन्यवाद करना चाहूँगी, जिनका सहयोग और पूरा समर्थन हमें इस कार्यक्रम को आयोजित करने में रहा है. इसके बाद हमारे जो आज के माननीय वक्ता हैं डॉक्टर योगेश सोजी जी एंड डॉक्टर सोनल सक्सेना जी का भी मैं विशेष आभार व्यक्त करना चाहूँगी, जिन्होंने एंटीमाइक्रोबियल रेजिस्टेंस की जागरूकता के संबंध में हमें मार्गदर्शन दिया और इस महत्वपूर्ण क्षेत्र में अपनी जानकारी हमसे साझा की है. हम आगे भी इनसे सहयोग की अपेक्षा करते हैं. लास्ट बट नॉट द लीस्ट, डॉक्टर रितिका और उनकी पूरी टीम की भी मैं सराहना करना चाहूँगी, जिन्होंने इस कार्यक्रम के आयोजन में कठिन परिश्रम और समर्पण दिखाया है. इनके परिश्रम या इनके प्रयास और इनके थिंकिंग की वजह से कि हमें ये करना चाहिए, क्योंकि ये अपकमिंग प्रॉब्लम है, हम आज सभी साथ हैं, उस कार्यक्रम का हिस्सा बन पाए हैं. और सबसे ज्यादा महत्वपूर्ण किसी भी वर्कशॉप किसी के लिए हमारे श्रोतागण होते हैं, जिन्होंने इस वर्कशॉप में पार्टिसिपेट किया, अपनी उपस्थिति दर्ज की, लाइव भी जो हम लोग जुड़ पा रहे हैं, हमारे इंस्टीट्यूट स्टूडेंट्स के जो हमारे अधिकारीगण हैं, उन सभी का भी मैं धन्यवाद करती हूँ. इस सभी के साथ आप सभी को मेरा धन्यवाद और शुभकामनाएं. थैंक यू.
Thank you, ma'am. Thank you for facilitating and convening this one-day awareness MR. I think before we break for lunch, we'll have a group photograph with our eminent speakers. So let's all gather for a group photograph first. We'll be resuming with the competitions after the lunch break. So because the speakers had to leave a little early, so we had done the sessions early. So we'll resume for the competitions after the lunch. We'll gather here for a group photograph.
छोटा पड़ गया
quiz competition. I would request the judges for this contest, Dr. Reenu Mittal and Dr. Prita Mehra, to please come over the dais. She's coming. And for all the participants who participated in the quiz today, all the very best. We have the judge for the first contest quiz competition, Dr. Reenu Mittal. So the quiz will be coordinated by Dr. Archana and Dr. Supriya. So over to them.
Hello.
We would request the team members to please introduce themselves. Team 1.
Dr. Ashana, Dr. Sana Sharan. Team 1.
Team 2.
अरे और अभी तो आवाज निकल रही है तो नाम सुन लो डॉक्टर हटीर उसके बाद तो निकलेगी नहीं शायद
Dr. Rani Chatterjee. Dr. Purnima Rani. We are team 3. Team 3. Team 6.
Team 3, please introduce yourself.
Team 3, Dr. Dishant.
Dr. Pooja.
Dr. Babita. Team 4. Dr. Suhana. Dr. Charu.
Dr. Shruti.
Very good. And we have the last team.
Team 5. Dr. Ekta. Dr. Himani and Dr. Sana. She's coming in 5 minutes. Namas.
So I think Dr. Archana will brief you the rules about the quiz.
बंद ही होगा, उस पर छोड़ दो अभी, है ना? रिकॉर्डिंग बंद कर दो। कुछ दिखेगा तो यानी वाइड ही चलेगा कैमरा। हाँ देखिएगा नहीं, वो आवाज़ तो नहीं पर मैंने कभी लाइव के लिए एक बार पूछ लेता हूँ।
एकदम आपके पीछे जो है ना, प्लीज आप एकदम छोड़ दो।
So we will be having 3 rounds. Each round will have 2 questions for each team. So there will be no passing of questions for every team. There will be 2 questions in each round. Okay, whichever question is asked.
बराबर ना, आवाज़ बराबर आएगी ना? कैमरा तो वाइड ही कोई बात नहीं। अब कैमरा ना आपको पता शिफ्ट करना पड़ेगा, फ्रंट पे करना पड़ेगा, कोई नहीं सकता। तो ऐसा तो घूम तो जाएगा, जैसे वो पार्टी बोलेगी, ये बोलेंगे, वो बोलेंगे, घुमाएंगे। सेट कर लो अपने। पैकअप